If we take a look around us, we can see how there are many aspects of fluorescence everywhere. My name is Joshua Alexander.
My name is Avinaya Kalivupan.
And my name is Shaba Sandhu.
We will start off by talking about how fluorescence can be seen or used in the real world along with the science world.
It is being used to help make our lives easier, whether that is done by saving lives or a matter of saving time.
Before we go further into detail on how fluorescence is used in the real world or science world, we'll get started by sharing our knowledge on fluorescence molecules.
What are fluorescence molecules? Fluorescence molecules absorb light energy of a particular frequency and discharge light at a longer wavelength. The retained wavelength energy moves effectively and time before outflow rely upon both fluorescence molecule structure and its chemical environment. As the molecule in its energized state collaborates with encompassing particles, the particle molecules are also known as fluoroprobes. Fluorescent molecules are used to stain tissue, cells, or a material in a variety of analytical ways. A fluorescent molecule is a particle with a fluorescence properties. The molecule assimilate photons and emit photons of lower energy. Consequently, these molecules are chemically diverse, and the electron of a fluorescent molecule consume photon of a excitation light. It helps to raise the energy level of electron to a energized states. The frequencies emitted from the fluorescent molecules are dependent on the molecule. Photons are emitted, transfer less energy, and have a longer wavelength. Fluorescence molecules have been used around 100 years in biological research and development. Fluorescence molecules are divided into three groups. Number one, organic dyes. Organic dyes are described as emanations starting from optical change delocalized over entire chromophore or from intermolecular change move transitions. The small size of these fluors is the advantage over natural fluorescence molecules for bioconjunction system. Since they can be cross-linked to macromolecules like antibodies, biotin, avidin without disrupting legitimate organic capability. A wide assortment of dyes with trademark emitted and emission spectra and idea quantum yields and fluorescence application excitation coefficients are commercially available. Number two, biological fluoroprobes. The advantage of these fluoroprobes is that articulation plasmids can be brought into one or the another microorganism cells into life forms. To drive articulation of fluoroprobe, entire alone or melded protein of the interested with interested regards to the natural cycle is considered. The size of fluorescent proteins can be changed according to natural capability of the cellular protein in which the fluoroprobe is used, and organic fluorescent molecules don't commonly give the degree of photostability and awareness presented by the manufactured fluorescent colors. Number three, quantum dots. Quantum dots have been increasing since 1980s as they are being used in fluorescence applications in biological research. Quantum dots are nanocrystals with distinctive chemical properties that can give tight command over the overworldly attributes of the fluor. The quantum dots are nano-sized semiconductors that, when energized, produce fluorescence at a frequency in view of the size of the molecule. More modest quantum dots transmit higher energy than enormous quantum spots, and subsequently the radiated lights move from blue to red as the size of nanocrystal increments. Mm, Next, next. How fluorescence is used in DNA. In what ways DNA can be detected through fluorescence? DNA can be detected through fluorescence in two ways. Number one, fluorescent molecule detect amount of specific double-stranded DNA sequence while fluorescent dyes detect only one amount of double-stranded DNA. The fluorescent dye-based method uses dye that emits fluorescence when incorporating itself into double-stranded DNA. How can DNA be detected through fluorescence? The DNA can be detected through fluorescence by using the most commonly dye used for fluorescent detection is CyBr green. CyBr green dye function as an intercalating agent that emit detectable fluorescence when bound to double-stranded DNA. As PCR progresses and the number of double-stranded DNA increases, the the signal intensity increases. A post PCR melt curve analysis enables the detection of non-PCR. They are all melted at a different temperature. The fluorescence observed is proportional to the amount of DNA present. What are the benefits of detecting DNA through fluorescence? In this method, we don't need the calculation by ourselves. The fluorometers calculate the concentration for us. In fact, it is fast and easy to use. Thank you.
There are many fluorescent aspects in the science world, one being fluorescence guided technology. This technology was first used in 1948 when neurosurgeons used intravenous fluorescein in order to aid neurosurgery. The use of fluorescence guided technology to illuminate cancer cells, tumors, nerves, and other anatomical features and has shown to be incredibly effective. Because of the accuracy created by this technology's glowing features, doctors are better equipped to treat patients with complex alignments. Fluorescence guided technology is frequently used during procedures on the parathyroid glands, esophagus and stomach, colon, liver, kidney, breast, and gynecology. The process of this procedure is started off by a patient being given a blood injection of an FDA approved dye. The most common dye used in this procedure is iocyanine green. This causes the dye to bind to blood proteins and travel throughout the patient's body. Then, in order to better observe the tumor or cell, sophisticated cameras and lights are used, leading to a more precise surgery. In this diagram, we can see how the visible light is being shone onto the target along with the LED. This is done after the contrast agent, or also known as the dye, is injected into the patient's bloodstream. We can also see how there's a camera pointed downwards, taking real life footage of the procedure that is being done. This is an image taken before the use of the fluorescence guided technology, and this is an image taken after the use of fluorescence guided technology. And you can see the difference of the colors that is being shown. Fluorescence guided surgery has many advantages, some of which include how it produces excellent contrast pictures, how there is low cost, avoids radiation, safe, and easy to use. The technology employed in this technique is advantageous since it can create photograph of any type of tissue, including those that are not immediately visible to the naked eye. This enables surgeons to be more precise in their work. Another well-known feature of fluorescence-guided surgery is GPS light tracking, which give medical professionals far more precise and thorough information. Another aspect of how fluorescence is used in the science world can be seen through the fluorescence-assisted caries excavation. Dentists frequently employ the FACE technique, also known as fluorescence-assisted caries excavation, to quickly identify any cavities or caries that might be present. The conventional visual tactile method is the other one utilized to find any caries or cavities. It was established in 2002 and has been in use ever since. Fluorescence assisted Cary's excavation is preferable to this method because it is more exact and accurate. This method uses a mirror and probe and is less accurate. The process of this procedure is done by using a violet blue light to illuminate the teeth during this technique, and a filter is used to block the reflection excitation light while allowing longer wavelengths to pass. Using a 530 nanometer filter allows yellow, orange, and red fluorescence to pass, which is what is needed to detect caries and cavities. When carious dentine is present, the orange to red region will glow, with red denoting profound dental caries and orange denoting less severe dental caries. In this diagram, we can see how there is a light being shone onto the tooth and a 500 nanometer filter close to the eye, differentiating the yellow, orange, and red fluorescences. In these two images, we can see how the left one does not use the fluorescence-assisted caries excavation, and this is shown how the right image has a red spot which represents the caries.
What is fluorescent ink? Fluorescent ink has pigments that are able to absorb UV ultraviolet energy. When viewing through a human eye, it appears in a bright glowing color. Fluorescent ink emits electromagnetic radiation. How is fluorescent ink used in the real world? Throughout the world, fluorescent ink is used in many different things such as highlighters. The ink in all highlighters is fluorescent ink. The reason the main ingredient in highlighters is fluorescein is because they are mostly brightening than any other chemical compound. When comparing any other ink, fluorescent ink absorbs and selects more light. What other products use fluorescent ink? Other products such as accessories, household decorative products, fluorescent fountain pens, glass tip pens all have fluorescent ink in them. What is fluorescent fabric? Fluorescent fabric is also known as polyester cotton interwoven fabric, polyester clover cotton. The fabric in the front is dyed with bright colors while the back is high-strength cotton, meaning it's strong and comfortable. The cloth is 2x fabric that has bright colors and fluorescence that reach the EN471 standard. This fabric is designed to glow and is created by special dyes. Its main usage is to be a safety protection for children, firemen, road workers, policemen, etc. How is fluorescent fabric used in the real world? Fluorescent fabric can be found in various things around the world. Its main use is to help people be safe when they are walking at night or working at night. For example, we have all seen the construction vest construction workers use. They wear it throughout the day but is mostly visible at night. It's used to help drivers be more aware that people are working. Almost all construction vests are made of fluorescent fabric which is designed to reflect light. When any light hits the vest, it reflects in all directions, making the person wearing it more visible and drivers more aware. Fluorescent blaze. Fluorescent blaze is similar to fluorescent fabric. It is also known as safety orange. The color of it is more darker and it has a more reddish tone. It's mainly used on hunting gear and road signs. It also helps drivers and equipment officers have a clear chance of seeing construction workers throughout the day and night. What is fluorescent powder? Fluorescent powder is bright colored powders. There are 5 main colors: fluor red, fluor green, fluor pink, fluor yellow, and fluor orange. It is mainly used as a fluorescent fingerprint powder. Many police/criminal investigators use this to help find fingerprints of criminals. Fluorescent powder's main use is that it is designed to help leave an impression on the fingerprint of different colored surfaces. A feather duster is used to apply the powder. They use a UV black light to help view the fluorescent better.
I hope you all got some knowledge on what fluorescent molecules are.
And how they are used in the science and real world.
Thank you for giving us your time for listening.